all from the same plant parts. This is quite interesting as well. You've collected something, you've got your new species, you've got your fungus, and it's always good to try to go ahead and check what are the different types of species, what are the different types of strains that have all possibly have been collected from the same host. This will help you to make some comparison as well. Sometimes you might go ahead and check whether people have recovered same fungi from tropical regions, others from temperate regions. Sometimes when I read some new species paper, I find that the descriptions and the discussion is just too simple. And there are sufficient information in the literature where those taxonomists could have gone and retrieved those information and explain to an audience about what's happening across those species. Something that might be of interest as well is the difference in lifestyle. What I mean here is that if you've recovered your species as a pathogen, could similar species exist as endophyte? Could it exist as a saprobe as well? So again, sometimes you can make some comparisons as well. It might be possible that you've recovered yours as a saprobe and somebody else recovered a taxon from a plant which is diseased. So you can try to make an association. If morphologically they are similar, if genetically they are similar, and you came across a similar species as well, so it tells you a little bit that the same taxon can exist possibly as an endophyte or a pathogen, and then exist as a saprobe as well. In another paragraph, what you can do as well, you can briefly reinstate how your new species exhibit morphological characters that mostly resemble other species, which is quite important. Usually, whenever you identify your species, you rely on similarities of morphological characters with other species. Then you should say that my species has been identified as X because they have these characters. But if you are going ahead and describe a new species, then you should be able to mention as well how other mycologists will be able to identify your new species precisely. It should have something in there for it to be new. You should provide evidence as to why you think your species is a new one. So then you cite it there, okay, these are my species. It is similar to others in terms of X, Y, Z. However, upon proper morphological examination, we notice that there are Y, Z there that makes it different from all other known species. Ma'am, clear so far? Anybody sleepy? Need a coffee? Not yet? Okay. Now we've talked a little bit about what? About our morphological characterization. And one of the reasons why we go ahead with our phylogeny is to support our morphological taxonomy to a certain extent and try to place our species in a natural framework. Once you are done with your morphology, then you back it up with your phylogenetic evaluation. You might have your different trees. You pick one of them if you want to, or you can use both of them for phylogenetic inferences. You can state something like our molecular data supports herein the establishment of X, which could be your new species, as it is phylogenetically distinct with high statistical support from other related species sample. Then you put your figure in there. So when you put other related species sample, you mention what are those species there. For example, my species Y clusters with species X with high bootstrap support. It is also related to the other species Z in terms of our ITS or beta-tubulin DNA sequences. Then what you need to do is you try to explain why you think your species are mostly related to those species in here. You try to mention about the phylogeny and you back it up with your morphological data as well. I've explained before how you can try to map your characters on a tree and give people further information. So the same scenario applies in here. Pairwise differences between species and strains. This is where, again, you can try to mention about whether there is any genetic divergence, what are the different transition and transversions there, how similar or how different your species are as compared to other species. If you read that famous paper, Juan and Hyde, in 2016, which was published in Microsphere, you will see people are citing that everywhere now. So again, I'll ask you guys, go and read that paper a little bit. Again, what's interesting again when you talk about any new species is apomorphs. I explained before what is an apomorph. Under most circumstances, whenever you have a new species, you want to try to find where is that apomorph. That is, where is that new or novel characters that possibly makes my species different from other species. And this might be a phylogenetically significant marker as well. Sometimes when you do your phylogeny, you can see that your species goes in an independent lineage. That is, all other species are here, but yours take a diversion and it goes somewhere down there. And it's being supported. What you need to look for is there any types of characters that possibly could be used to identify specific taxon in there. It might also give a clue that that particular character could be very important in segregating species. We've talked about that yesterday. For example, we talked about how spore ornamentation can be very helpful in identifying species. So you could explore such an opportunity in your tree as well. But you should be careful about the number of species you include in your taxon sampling. Now, sometimes you might be having two species that are very similar. It does happen. So what you do is you state on what grounds you would justify your new taxa. If you have two species that are morphologically similar, that does not mean that one of them can't be a new species. It can be, but you've got to give a scientific reason as to why you think it is a new species. For example, despite the apparent similarities in ascomatal features, our new taxon lacks pseudoparaphytes and its spore septation is different. So this gives you an idea about how sometimes you can try to look for similarities and differences in your morphology and capitalize on such criteria to establish a new species. Sometimes you might see that you need to check for the asexual state or maybe vice versa. But sometimes you might have a problem. Let's say if you want to identify a new species and you got it from cultures and you got it from the asexual state, but most of its phylogenetic members are sexual fungi, then you will not be able to make a good morphological comparison. So this is where you rely on your phylogeny to state why my species is different. Sanju, okay? Do a good board today.